Have you ever wondered what it would be like to see inside your own cells? I mean, to literally send tiny glowing messengers in to deliver medicine exactly where it needs to go, and then watch them as they do their job? It sounds like something straight out of a sci-fi movie, but believe it or not, it's actually happening. So let's take a look at how these incredible glowing particles work. Right, take a look at this. You are not looking at special effects. This is a real image of human immune cells, and those brilliant glowing spots? Those are microscopic silicon particles that the cells have literally eaten. This is a huge deal, and it could totally change how we track and treat diseases on a cellular level. So why is this so important? Well, for a long time now, scientists have been trying to find the perfect little vehicle to carry drugs or vaccines right into our cells. But this tiny courier has to be a whole lot of things at once. It has to be safe, it has to be biodegradable, and, this is the tricky part, we need to be able to follow it. We need to track it as it navigates the incredibly complex world inside our bodies. And that's where our hero material comes in, porous silicon. I mean, this stuff is just amazing. It's made from the same element you find in computer chips, but it's engineered to be more like a tiny sponge. Our bodies don't reject it, it's biocompatible. It dissolves over time into something totally harmless, so it's biodegradable. And here's the best part. It comes with its own built-in GPS. It naturally glows with this beautiful, bright orange light. Here's what one of them looks like up close. We're talking seriously small, usually between 1 and 10 micrometers. That's about the size of a single red blood cell. And you can see that rough, sponge-like texture, which gives it a huge surface area for carrying whatever medical cargo we want to load it up with. But you know how it is in science? It's never quite that easy. Researchers hit a major snag, a huge roadblock that basically threatened to switch off the silicon's best feature. It almost made that glowing tracker totally useless. See, for these particles to actually carry anything, they need little chemical hooks on their surface to grab onto the drug molecules. And one of the best kinds of hooks for the job are something called amine groups. They are fantastic for attaching all sorts of biological cargo. Sounds perfect, right? You just add the hooks, load them up with medicine, and follow that beautiful orange glow. So, what's the catch? And here it is. This was the showstopper. The very amine groups they needed to attach the medicine are famous for quenching silicon's glow. And by quenching, I mean they turn it off, completely. It was a classic catch-22. If you make the particles useful for carrying drugs, they become invisible. If you keep them visible, they can't carry anything. But a team of scientists came up with this incredibly elegant solution. It was a chemical workaround that didn't just solve the problem, it accidentally unlocked an entirely new superpower for these particles. So, instead of attaching those amine hooks directly to the silicon, they did this clever two-step dance. First, they coated the silicon in a protective shell of something called acrylic acid. Then, they attached the amine hooks to that outer shell. The result? The amines could still do their job, but they never actually touched the glowing silicon surface. Genius. And just look at the result. On the left, that's the particle with its normal orange glow. But on the right, after they did the two-step fix, wow. Not only did they save the orange glow, but a brand new, totally unexpected blue light appeared. By solving one problem, they accidentally created a dual-color particle. And the researchers themselves confirmed it worked perfectly. They wrote that the two-step procedure didn't cause any quenching because, as they put it, the silicon surface was well protected. The fix worked. It was a home run. Okay, so now we have a particle that glows in two colors. But here's the really cool part. These two lights are totally different, and that difference is what makes this so powerful. The best way to think about it is like this. It's like having a glow-in-the-dark toy and a camera flash built into the very same tiny particle. Let's break that down. The original orange light, that comes from deep inside the silicon core. And its lifetime, how long it glows for, is really long. We're talking microseconds. It's just like a glow-in-the-dark star you'd stick on your ceiling. It just keeps on shining. But that new blue light is different. It comes from tiny defects on that new chemical surface they added. And its lifetime is incredibly short, just nanoseconds. It's a quick, intense burst of light, just like a camera flash. Now, why does that lifetime matter so much? Well, a living cell is a very noisy place, visually speaking. There's all sorts of stuff in there giving off little brief flickers of light. But because that orange glow is so long-lasting, it stands out from all that background chatter. It's easy to spot, even in a crowd.
And that super fast blue flash, that's the smoking gun. Its nanosecond lifetime is proof positive that it's coming from a completely different source, those surface defects. This gives the particle a unique signature. It's a 2-speed, 2-color barcode that screams, "Hey, it's me," and confirms its identity.
So, the theory is fantastic, right? But here's the moment of truth. Does it actually work inside a real living biological system? Let's take a look at these dual-color particles inside actual human immune cells. And there it is. This is it, in action. These are human dendritic cells. They're a key part of our immune system. They've gobbled up the particles, and you can see them glowing brightly inside. By using different filters, the scientists can see the exact same particle light up in both the blue/green channel and the orange/red channel. It works.
This close-up really drives the point home. We are zoomed in on one single particle inside a cell, and you can see it glowing in the green range on the top left and in the red range on the top right. Having that 2-channel confirmation makes tracking these things incredibly precise. There's no mistaking it for something else.
Okay, but here's maybe the most important question of all. Is it safe? I mean, none of this matters if the particles are toxic, but the results here are fantastic. This graph shows that even at really high doses, over 85% of the cells were still alive and perfectly healthy. They are, for all intents and purposes, non-toxic. So let's put it all together. We have a particle that's safe, that we can track with incredible precision. What does this actually mean for the future of medicine? Well, what we've got is a platform that is non-toxic and breaks down safely. It lights itself up with its own unique 2-color signature. That means you don't need to add any other fluorescent tags, which can sometimes be harmful. And it's been proven to work perfectly for tracking things inside our own immune cells. The potential here is just massive. You're looking at a powerful new platform for creating things like nanovaccines that can target very specific cells, or for tracking exactly how a drug is being delivered in the body with a level of precision we've just never had before. It really makes you wonder, doesn't it? With this power to glow in 2 different colors, to carry medicine, and to safely track their own journey deep inside our bodies, could these amazing little particles be the key that finally unlocks the door to the next generation of truly personalized medicine?